Welcome to The Explainer. Today, we're diving into something totally fascinating. We're not just looking at the cell as some basic biological unit. No, we're going to explore it as an absolute masterpiece of structural engineering. So, get ready, because we're about to see the microscopic world like you've literally never seen it before.
Okay, let's dive right into this. Have you ever looked up at a giant tree, maybe a massive oak or a towering redwood, and just wondered, how on earth do plants stand hundreds of feet tall, bearing absolute tons of weight against gravity without a single bone in their body? I mean, humans, animals, we rely on a complex internal skeleton to stay upright, right? But plants, they don't have that. The secret to this massive structural puzzle actually lies entirely in the microscopic fortifications of the cell itself.
So here's our game plan for today. Grab your notebooks because this is going to be a data-dense deep dive. We'll start with the anatomy of the cell, then look at the ultimate cellular fortress. Third, we'll check out wall diversity across species. Fourth, the architecture of wall layers, and we'll wrap up with the vital cell wall functions.
All right, starting with Section 1, Anatomy of the cell, the baseline blueprint. Let's get our foundation set before we zoom into the outer defenses.
Now, what's really interesting about this slide is how it perfectly visualizes the fundamental difference between squishy animal cells and the rigid, fortress-like structure of plant cells. On the left, you've got a typical animal cell, kind of like the ones making up the inside of your cheek. It's soft, it's highly flexible, and it's bounded only by a super thin cell membrane. But look at the plant cell on the right. It's got this geometric, almost block-like shape, right? That's because it's completely encased in a tough, rigid cell wall. And inside, there's this large central vacuole filled with water that's literally pushing outward against that wall, keeping the whole cell firm and upright. That combination right there, that's what gives plants their structural integrity.
Moving on to Section 2, the ultimate cellular fortress, membrane versus wall.
Okay, let's really slow down and break this down because it's an absolutely vital concept. On the left, we've got the cell membrane. Now, this is a living structure. It is extremely thin. We're talking less than 10 nanometers thick, and it's semi-permeable. Basically, it acts like a highly selective security guard, choosing exactly what gets in and out. Plus, the cell membrane is found in all cells, no exceptions. Now, contrast that with the cell wall on the right. Believe it or not, the cell wall is actually dead. Yeah, it's this non-living armor secreted by the cell. It's highly rigid and it's fully permeable, meaning it doesn't filter molecules based on size the same selective way the membrane does. Instead, it acts as this thick structural protection. And crucially, you won't find it in animals. It provides this extra structural fortification specifically to plants, fungi, and bacteria.
Which brings us to Section 3, wall diversity across species, building materials, because different biological kingdoms use entirely different chemical bricks and mortar.
All right, get your notebooks ready, maybe hit pause if you need to, because this is highly testable data. Look at the incredible diversity here. Nature is just an amazing architect, using complex sugars and proteins to build these barriers. For the Monera kingdom, our bacteria, the wall is primarily constructed from peptidoglycan, which is also called mucopeptide. Fungi, like mushrooms and yeast, well, they use a completely different material called chitin, alongside fungal cellulose. It's incredibly tough stuff. Algae rely on a mix of cellulose, galactans, and mannans. And finally, the plantee kingdom builds these incredibly resilient walls using a matrix of cellulose, hemicellulose, pectin, and this tough woody substance called lignin.
And this brilliantly illustrates that the diversity doesn't just stop at the kingdom level. We see massive structural differences even within a single kingdom. Check out these two different bacterial fortresses, gram-positive versus gram-negative bacteria. Notice how gram-positive bacteria have a very thick, layered wall, with peptidoglycan making up an astounding 80% of its chemical composition, right alongside teichoic acid. But look over at the gram-negative bacteria. They have a much thinner wall, only 3 to 12% peptidoglycan, and absolutely zero teichoic acid. But hey, don't think they're weaker for it. They actually compensate for this thinner wall by possessing an entirely extra outer membrane that the gram-positive bacteria completely lack. It's just a completely different evolutionary strategy for defense.
Let's zoom into the plant kingdom, specifically now with Section 4, architecture of wall layers inside the plant fortress.
You can really think of a plant cell wall like a high-tech construction project being built from the outside in. First, the cells lay down the foundation, right? So step 1 is the middle lamella. Once that's established, the growing cell builds step 2, the primary wall, located just inside that lamella. And finally, when the cell reaches maturity and really needs to harden its permanent defenses, it constructs step 3, the secondary wall.
And get this, the secondary wall isn't just one solid block. It's laminated into 3 distinct, incredibly strong sublayers that we call S1, S2, and S3.
Let's look a little closer at that first layer. The middle lamella is basically the biological mortar that glues the cellular city together. It's the outermost, thin, amorphous layer that gets deposited right when the cell divides, a process known as cytokinesis. So what's this mortar actually made of? Well, it is specifically made of calcium and magnesium pectate. Those pectin compounds act exactly like sticky cement, binding 2 adjacent plant cells tightly together so they can actually form a unified tissue.
Now, once those cells are glued together, we see a really fascinating contrast in the inner walls. The primary wall is built first, inner to the lamella. Think of it kind of like flexible scaffolding. It's thin, it's elastic, and it's made of a loose network of cellulose microfibrils floating in this gel matrix of pectin and hemicellulose. This elasticity is absolutely crucial because it allows the young plant cell to expand and grow. But, and here's the cool part, once the cell stops growing, it lays down the secondary wall. This is the permanent lignified concrete of the mature cell. It's thick, completely inelastic, made of dense cellulose and lignin, and heavily laminated into those S1, S2, and S3 sublayers we talked about, maximizing structural strength and locking in the final shape of the cell.
Let's move on and see how this builds. Because obviously a fortress without any doors or gates is basically just a tomb, right? The cell still needs to trade resources and communicate. And that is exactly where plasmodesmata and pits come into play. Plasmodesmata are actual microscopic channels that connect the living protoplasm of adjacent cells. Pits, on the other hand, are uneven, unthickened regions located right in that tough secondary wall. They essentially act as permeable gateways, allowing vital water, nutrients, and chemical signals to easily pass through those heavily fortified borders. Bringing us to our final section, Section 5, Vital Cell Wall Functions, the Defensive Protocol. Let's summarize the operational purpose of all this complex architecture.
So, the crucial point is, what is the ultimate operational purpose of this entire cellular fortress? I'd actually recommend you take a screenshot or jot down the specific slide for your study records. First, it offers a definite shape and rigidity to the cell, which is exactly why plants can stand so tall. Second, it serves as a physical barrier for several molecules. Third, it provides immense protection to the delicate internal living protoplasm against any mechanical injury. Fourth, and honestly, this is just an elegant piece of engineering. It prevents the cell from bursting by maintaining osmotic pressure when water rushes into that central vacuole. And finally, it acts as a primary frontline mechanism of defense against invading pathogens.
As we wrap up this explainer, I really want to leave you with this provocative thought. How might human architecture and engineering learn from the dynamic, microscopic, laminated defenses of the cell wall? Think about it. From self-assembling mortars to multi-layered flexible to rigid laminations that allow for both growth and permanent strength, nature has been perfecting structural engineering for literally billions of years. Keep wondering about the microscopic world around you, keep taking great notes, and I will see you on the next explainer.